Okay, so here's a very elegant experiment that you could do, which will now prove to you that our instinct is wrong. Why was our instinct wrong? Because we ignored evolutionary biology, right? We ignored the actual body plan of the animal. So here we have an electric fish. He's been chemically manipulated so that the electric organ, the nerve muscle synapse at the electric organ is no longer working, so the animal can't actually produce his own electric organ discharge, but the nervous system is still sending volleys down to the electric organ, and you can measure what the animal's trying to do. You can gently put one electrode in the mouth, another near the tail, and stimulate and produce your own fictive electric organ discharge around the animal. And then you can put electrodes either in front and behind or side to side of the animal and create some sort of jamming stimulus. So a very elegant preparation. And now you do the experiment with that preparation where you put the animal in two compartments. You have a Vaseline seal, so there's only about 1% leakage of the signal from one chamber to the other. And you start producing giving various signals into the two chambers. Okay? So one thing you find right away that's quite fascinating, if you give some combination of SA and SB, the signals into the two chambers, and you put that same combination into the two chambers, you never get a jamming avoidance response. That is, the animal never attempts to try to move its own electric organ discharge as signaled by the nervous system, the reading of the nervous system. If you put some combination of SA and SB just to the head region or just to the tail region alone, you don't get a behavior from the animal. Okay? What you need, so what you need is a difference between the head region and the tail region that drives a change in behavior. That is, when the animal detects that difference, it will move its attempt to move its own electric organ discharge, that is the nervous system readout, will say higher or lower. And remarkably, and this is perhaps the most compelling, if the animal, you can read the volleys coming down the spine, right, spinal cord. So you can tell that the animal is, say, at 400 hertz. You can be producing an EOD, electric organ discharge, around the animal that's at 500 hertz. Now if you give the appropriate stimuli to create a jamming signal at, say, 502 hertz or 494 hertz, so the animal should go up or down, his nervous system will signal appropriately that he's going up or down, even though it's at 400 hertz. So the two signals are separated in very interesting ways. Right? The plot thickens. So they have an interesting solution, a very interesting solution. It's just not the straightforward one that we would think of from first principles in engineering. You can add two different signals, S2 and S3, to the original S1 and start to get, you can really play games and get the animal to paint doilies in this phase plane plot, and you get that behavior from the animals. It's really quite elegant. And you can predict breakdowns of these behaviors through the nonlinearities in the receptors and various aspects of the behavior. I don't have time to develop that in detail. So here's the concept that we're developing, that the animal isn't paying attention purely to the S1 signal the way we thought of it, but instead it's calculating differences on different parts of the body surface. Why would it do that? Because it has to calculate phase, you can calculate amplitude, no problem. But to calculate phase, you need a reference. What is zero phase here, right? And so what the animal is doing, because it doesn't have access to the EOD discharge itself, it's calculating the contamination, the signal at A relative to B, the signal at B relative to A, and the signal between lots of different parts of the body surface. A to B, B to A, and A and B can be all over the place. Okay? So now if we look in the phase plane, things have changed a little bit. So here is the amplitude of A as a function of the phase at A, but the animal can't actually calculate this. Here's the amplitude at B, relative to the phase at B, and the direction is the same. But now if you look at the amplitude difference and you calculate the phase difference, you start seeing you start seeing some of the complexity that the animal has to work with. And in fact, when you do this calculation, you'll see that A relative to B is telling you go down in frequency. B relative to A is telling you go up in frequency. So you may ask, what's the advantage of that? Well, the amplitude of this signal is smaller than that signal. So Walter Heiligenberg, he and his colleagues in his lab discovered this. Walter called this a noisy democracy. You have some people voting one way, other groups of people voting, or neurons voting another way, but there are more people voting, neurons voting the right way than the wrong way, so the animal does the right thing. Okay? So it's a pretty crazy scheme. Who would believe such a crazy story, except it's true. Okay, so let's now dive into the nervous system a little bit. That's about the behavior. Let's now dive into the nervous system and learn a little bit about what we need to know about to start our instruction in neuroscience, and we'll learn things about how brains are organized in general, and we'll also learn about it in relationship to this particular behavior. So Eigenmannia have a complicated nervous system. It follows the vertebrate body plan. This should not be a surprise because they're vertebrates. The parts have all these complicated names that are very strange to us. They're very strange to me. And that's not a surprise because when anatomists were first doing this, they didn't know about all these relationships. This only we've discovered later on. So they have strange names, and ultimately we discovered what the relationships are. Okay, no problem with that. That's about the basic organization. So all animals, except some groups of animals that we don't have time to talk about, have neurons. Okay, that's useful. That's a type of cell, right? A cell is an amazing thing. It's alive. All of life is in that one cell. That's extraordinary, right? So there are specialized cells in multicellular organisms that are neurons that communicate, that create a nervous system. And these neurons have inputs, which we call dendrites, because it's like a dendritic arbor, like the shape of a tree, that all come into one cell body. It's sometimes called a soma. And the cell body sends an output, which we call an axon, and the output of the cell is what carries information to other cells. So dendrites, cell body or soma, and axon. Input, central point of collecting the inputs, and then output. Okay? So the neurons are connected together. They form networks. There are different types of connections from one neuron to the next. Those connections are called synapses. Sorry for all the terminology, but that's what we have to deal with. And there are two types of synapses in general. Some are chemical in nature. Some just pass the electrical current. We don't have to worry too much about that, but you should know. And the general idea, which I will challenge at the end of the next lecture, is that changes at the synapses, so-called plasticity of synapses, is the fundamental change that drives learning. Okay? So that's good to know. If we're going to build networks of the nervous system from biologically realistic neurons, we need to know about this stuff, right? So it makes sense to dig into it a little bit. So most neurons spike, or they have this sort of highly nonlinear behavior. The electrical potential inside the cell approaches some threshold level, and if it exceeds that threshold level, and especially if it exceeds that threshold level quickly enough, then the neuron produces a spike or action potential. And that signal, that spike or action potential, travels down the axon to the synapses at the ends of the axons where it interacts with other neurons. Okay. So here's an example of one extracellularly recorded spike. That means the electrode wasn't inside the cell, it was just close to the cell. And you see that the voltage goes up, it drops down, and then returns to baseline. The amplitude here is completely artifactual in an extracellular recording. It doesn't matter. The time course is a little bit more relevant. And the spike occurs over a millisecond. The whole thing might take several milliseconds to relax. Okay? And there are different types of neurons, excitatory and inhibitory ones. We'll get to that in a moment. Okay, so here's one observation. So if you look at the receptors along the body surface, they're organized, right? So the receptors along the head are either close to each other or further away, and then there are the receptors of the bodies that can be close to each other or further away, and so on and so forth. So there's a map of the body created by the physical structure of the body of the receptors. That map is preserved in the brain. We call that topography, right? Topographic organization. And this is true for the receptors of the fish, just like it is for the receptors in our eyes or our own body surface. So many of you may have heard of the term homunculus, the representation of the body surface in the brain, which can be stretched, right? The hands are much bigger in the representation in the brain, for instance, than the trunk. Well, there's also a fishunculus or a lectunculus or whatever in Eigenmannia. So there are these topographic organization. And now there are receptors. Okay, let's talk a little bit about those receptors. There are two types of receptors, of these high-frequency receptors. Those that detect time information. You see how this unit is firing always at the positive going zero crossing of S1 plus S2. And those that probabilistically, that's why they're called P units, increase their firing rate when the amplitude goes up, or some of them decrease their firing rate as the amplitude goes down. So this is a time signal, this is an amplitude signal, and it's already separated at the level of the receptor. And those are the two things that the animal's going to have to measure, right? So, and here you see recordings of so-called suprathreshold or spiking activity of a couple of these receptors. Okay, so you can now sum up, give a lot of sine waves, and sum up the activity of, say, a P unit or a T unit, and now you've got a rate code. You're no longer detecting what's happening on a trial-by-trial basis, which is the problem the nervous system has to solve, right? But you're making the assumption that the statistics are about, give you some valuable insight. The mean firing rates of the neurons give you some, or receptors in this case, give you some valuable insight. And so you now have a rate code. So this is a rate code, a representation of the likelihood, the P unit firing as a function of the phase of the beat cycle, and this is the firing of a T unit as a function of the phase of the beat cycle. And you can combine these two signals together and create the representation you'd expect to get. That's very satisfying and tells you that maybe you're making some progress here. If you look at the way the receptors project into the brain, you find out that one patch of receptors somewhere on the body surface projects to the first cells in the central nervous system. Okay? So the cell at the earliest stages of the central nervous system don't see the whole body. They see a small patch of it. So the cells in your retinal ganglia, the output cells of your retina, which are the ganglion cells, also project to cells that get input from a local region within the retina. So that means the nervous system is splitting up the larger receptive sensory field into small patches, which allows it to do calculations on those small patches, but also introduces artifacts that become visible in behavior and so on. Okay, so now let's look at how that convergence occurs, how we get this, these different types of synaptic inputs onto the cells through different patterns of convergence of receptors onto the cells, what sort of processing might go on. So I think for lack of time and because it's so much detail for you that may be new, I don't know that I need to go through the details, but if we just consider, for instance, the P receptors, they project onto two types of cells that they're formed, their morphology is different. So they're called basilar papillar or non-basilar papilla cells. It doesn't matter, except they're 1 and 2. They're called 1 and 2, okay? And one gets the inputs of the P receptors are excitatory synapses from the middle of the receptive field, and they project onto other neurons which produce inhibition onto these one cells, but they're at the edges of the receptive field. So you have excitation in the middle and inhibition on the outside. And the two type cells have inhibition in the middle and excitation on the outside. So this is called center-surround organization, an on-center and an off-surround, or an off-center and an on-surround. Okay? Now, why is that of interest to us? Well, let's look at the example in the visual system. So here you have on-center, off-surround, and off-center, on-surround cells in the visual system in your retina. Your retina has retinal ganglion cells that have this property. And if we now look at how we sum information, so the on-center, we'll discover a very fundamental phenomenon in the brain. So the on-center, we could think of it as input that sums, positively sums into the cell. And the surrounding inhibition, we can think of it as signals that, subtract from the signal in the cell. So this cell that sits somewhere here and receiving constant luminance across its whole receptive field has an output of 2, all right? Plus 4 and 2 times minus 1, okay? Now, as we shift that cell over, what we'll see is that at the edge of the change in contrast, the response will go down. On the other side of the edge, the response will go up, much higher, and then here it'll be constant again. Okay? So if we look at that across all the different cells, what we'll see is this. This is the physical stimulus. You take your instrument and measure across the stimulus, and you get this signal. This is the perceived stimulus. It has dips and peaks near the edges. This is contrast enhancement. This is lateral inhibition, which is what happens when you have center-surround organization in a topographically organized space. This is how the nervous system, all nervous systems do this. This was first discovered in a dumb animal, a limulus, a horseshoe crab, an animal that is very primitive. It's been around successfully for many hundreds of millions of years, has lots of different eyes, and it has lateral inhibition in its eyes. And we see lateral inhibition everywhere. So we see lateral inhibition all the time. So is this effect a potent effect?